Hello and welcome to the webinar today, titled Induction of Receptor Tyrosine Kinase Signaling by Sera Isolated from Patients with Lung Adenocarcinoma, brought to you by Oncology Central. My name is Roshane Vijayatunga and I am the Senior Editor for our Oncology portfolio at Future Science Group. I'm happy to be your host for today's event. We are delighted to have two speakers presenting for us today. Jeffrey A. Borgia, Associate Professor at Rush University Medical Center in Chicago, and Wenrong Li, a Principal Scientist at MILLIPLEX. Dr. Borgia has been at Rush University Medical Center for 15 years and is an Associate Professor in the Departments of Pathology and the Department of Cell and Molecular Medicine. Dr. Borgia also holds the positions of Director of the Rush Biomarker Development Core, as well as the Rush Translational Cancer Research Graduate Program. His research is focused on developing novel omics diagnostic tests for profiling patient specimens and providing mechanistic insights for pathophysiology. Dr. Borgia oversees the collection and annotation of patient specimens for translational research projects and ongoing clinical trials at the Rush University Cancer Center. Additionally, at the core, he and his team work on discovery and development of new novel biomarkers for use in diagnostics. Dr. Wenrong Li is a Principal Scientist at MILLIPLEX and has been working on biomarker immunoassay development there for 13 years. She has developed multiplex protein immunoassay panels for studying skin and bone metabolism, angiogenesis, neurodegenerative diseases, cell signaling pathways, immunology, and oncology. Her current work is focused on developing new biomarker assay tools for immuno-oncology research and cancer autoantibody profiling. Dr. Li has also worked with researchers in academia and pharma to develop custom biomarker panels and to advance science through research collaboration. We invite the audience to submit any questions you may have for our presenters throughout the webinar using the Q&A tool to the right-hand side of the screen. We will pose your questions to both speakers in a live Q&A session after the presentation if time permits, and then we'll follow up any outstanding questions offline. Before we proceed with our first speaker, we have a quick poll question for our audience today. The question is, are you familiar with or do you use multiplex assays based on Luminex XMAP technology? Your options are, yes, I use multiplex assay based on the XMAP technology. Option 2, yes, I am familiar with the technology, but I do not use the assays in my research. And option 3, no, I am not familiar with this technology. And if you could please click on the box that is relevant to you. Okay, and we'll just move to the results slide. And we can see that from the results, the majority of you are not familiar with this technology, which is great because hopefully you'll learn a lot from this webinar today. So thank you to all for answering this question. Thank you once again for joining us today. And I'll now hand over to Dr. Borgia to start today's presentation.
Thank you, Roshane. I'd like to thank everyone for signing in today to listen to me discuss some of our research interests and some of the results from a recent project that we had complete. And with that, I mean, our project, or rather our laboratory mission, is really to develop novel molecular diagnostics using the Luminex platform for the most part. But we're trying to develop new blood tests that we can use to help guide patient treatment for lung cancer patients. I have interacted with other groups too on other types of cancers, but for the most part, lung cancer is our primary research area. And from this, we'll look at everything from early detection through treatment selection for the patients with advanced stage disease. Lung cancer, just as a little primer on lung cancer, lung cancer is by far the #1 cause of cancer-related death worldwide. In the United States, it kills more people than the next 4 different carcinomas put together. That means including prostate, breast, colorectal, and pancreatic cancers. There's more deaths from lung cancer annually from those, or from lung cancer, than from those other ones put together. And so with that, the biggest issue is lung cancer tends to be very asymptomatic and impacts a lot of people. And with progressive stage, the survival time goes down dramatically. And it's only until recently there's been any sort of effective means of even remotely prolonging survival, but that requires treatment selection, which is the topic of what we're covering today. However, with early detection, comes better treatment options, and that's mainly surgery. And with that came the emergence of lung cancer screening programs to try to catch lung cancer at a much earlier stage where surgery is still an option. Most patients, when they become symptomatic, have the later stages, which have a very grim outcome. And treatment options are very limited with disease once it's disseminated. And so one of our lab missions is to try to catch it earlier so we can move them forward to surgery and hopefully get a cure that way. But if the disease has already taken hold, the best bet for us to do is to understand it better so we can design better treatment strategies and help select those strategies better, as I've already discussed, with a blood test or possibly some tissue-based test. The primary mechanism that we evaluate for that is really looking at phenotypic changes in these tumors as the disease becomes more aggressive and starts progressing with the patient. And so as a quick primer, looking at the figure on the left, when we describe stages, we really talk about how metastatic is the tumor becoming and how invasive. And so the stage 1 tumors are just a solitary nodule, and those for the most part can be relatively easily excised through surgery, and treatment outcomes for those are the best of all the different stages. But once it starts progressing to these local lymph nodes that are shown here, then the stage number starts going up to 2 and 3, depending on how far it goes. Once it gets outside of the diagram here, now we have stage 4 disease, and that's disseminated disease. And that is much harder to treat, and so that's where the outcomes definitely fall off. The mechanism underlying all of this is the leading mechanism, hypothesis I should say, is EMT. That is, there's phenotypic changes that happen in these cells that permit them to start spreading, that allows the tumor cells to break off from the primary tumor and start invading surrounding tissues, travel through the lymphatic or the circulatory system, and colonize other tissues. There's a fair amount known about this program already, EMT that is, such that there's a series of different tissue-based markers which are currently used to try to characterize these. And indeed, we've used some of these markers ourselves in the study that we recently published. That is, in a total of about 100 surgical cases, we went through and we looked to see for EMT, which are the top 2 markers here. E-cadherin is a marker for the epithelial phenotype, whereas vimentin is a marker for the mesenchymal phenotype. Again, the mesenchymal phenotype is associated with metastatic progression. So when you see the brown, that means there's positive immunostaining for that marker. So E-cadherin, which is in this case the better marker, is primarily only seen in the panels on the left. These are patients that don't have nodal metastases. And so at the top there, you see the T, N, and the M's. So the T describes the size of the primary tumor. The N describes whether or not there's nodal metastases, and the M is metastatic. That's disseminated metastases. And so on the left, you see that there's much higher prevalence of E-cadherin staining when there's no metastasis versus when there is metastasis in the panel on the right, versus the opposite is true for vimentin. So there's already some known markers, and indeed, these have prognostic significances. So when we look at a ratio of those two, and so we're doing here a ratio of vimentin to E-cadherin, which is in effect a ratio of how mesenchymal is the tumor. The higher that number, the worse the outcome. And so the red number is, or the red line here in these Kaplan-Meier plots are the patients that have the higher levels versus the black. And so this is a survival curve, basically, so for both of these, just as a quick primer. And the x-axis is time. And so what would be ideal is you see a line go straight across. That means no one is having a recurrence and no one is dying from the disease. But as time goes on in these, there's attrition, and that causes the line to drop down. Okay, moving on. So EMT, as we've just shown, is pretty well entrenched in metastatic progression, but it's also very involved in many of the other hallmarks of cancer that are, very well described in this very seminal paper by Henhan and Weinberg. And our goal, as I mentioned before, is that we really want to try to identify a secretome for many of these different hallmarks, and in the case that we're talking today about EMT, That is, we want to have a series of markers that we can sample through the blood that will indicate what's going on phenotypically with the primary tumor. And our hypothesis for this, of course, is that there's, these markers, if we can find mesenchymal markers in the blood, then that gives us an indication that there's a negative prognostic situation happening. But of course, there's not a whole lot known about this currently. So one of the things we did is, in collaboration with the Cancer Genome Atlas, we decided to do a couple different analyses looking at the initial sets of reposited data that they have published. And so with this, we did two different comparisons. One comparison was looking at the incidence of disease recurrence, and the other is looking specifically at what I showed you a moment ago, which is looking at the difference between patients without nodal involvement versus those that do have lymph node involvement of their disease. And recurrence, as it says in the slide here, is, in essence, we're considering that to be strictly a case where there's metastatic progression, but it's too small to be detected through current clinical mechanisms of staging the patients. And so it's a positive lymph node, just we can't see that it's positive just yet. All right, so from this collaboration, we're able to do two different analyses, as I just said. So the first one for recurrence, we're able to identify a series of circulating markers. That is, these are markers that are predicted to be secreted at the RNA level, or markers that are different at the RNA level that bioinformatically are predicted to be secreted. These secretions, these secreted markers are now candidates for us to evaluate in the future. And we have a similar set of lists for that for looking at nodal status. These are things that are currently definitely in our list to be examined going forward. But before we're going to do that, we had a couple questions that really arose that we wanted to answer before we took that step. And one of these is that there's numerous studies, including like some that we've published, where there's direct associations between circulating levels of different protein biomarkers and the outcomes of the patients. But the mechanistic significance of those are, at this point, really just we're supposing that these are having an impact. There's not a direct line of evidence showing that, if we're measuring a cytokine that's measuring, is that actually signaling the tumor cells? And so what we wanted to do is try to unravel this a little bit, try to unpack that to see, can we see if that is a direct sort of effect? If you see higher levels of IGF-1, for example, does that mean that there's higher levels of IGF-1 signaling in one case versus another? Because there's all sorts of other factors that could potentially regulate that, including decoy receptors and other binding proteins, as well as partial ligand degradation that is possible to occur. And as a quick illustration of where this was not studied is in the case of the clinical trials involved in IGF-1R inhibitors, fidgetidumab. And in this case, the clinical trials in lung cancer did not go so well. And in fact, ultimately, they were discontinued, primarily, perhaps in my opinion, primarily because there was not a good way to select. There was no circulating biomarkers to select patients. That is, there was not a mechanistic understanding of what was going on with the patients that were having a response versus those that weren't, that needed to be explored. And that's part of what we really kind of specialize in, is trying to understand those, try to develop new biomarkers that have mechanistic impact. And as you might have guessed, our primary instrument for this is using a Luminex platform. And we have a FlexMax system in the lab that performs brilliantly for us. And so it gives us a very cost-effective way to evaluate our biomarkers. It's very sensitive, and of course, the throughput on it is phenomenal. So this is definitely our platform of choice for doing these type studies. It operates on the basic principles of a sandwich ELISA type assay, immunoassay, but provides the ability to plex, and so we don't have to use large amounts of sample, doesn't require a lot of manpower, and we can go through these studies pretty rapidly and get like very robust data in a short period of time. Again, so the topic that we wanted to study first had to do with receptor tyrosine kinase signaling. And of course, we were working together with MILLIPLEX on this, and we found and they were developing this kit which directly assays receptor tyrosine kinase activation for the members that are listed on this slide. And this would give us a means that we could directly measure whether or not we have activation of these ligands if we did an experiment where we're directly stimulating cells and then we studied the lysates of those cells. And I'll come back to that in a second. But paired up with that would be the idea that now we can characterize the serum before we go into that experiment for what are the circulating levels of the ligands, what are the levels of the decoy receptors that might be in there. And so combining those two ideas together, we're able to, in a single experiment, or a single set of experiments, I should say, evaluate what's the composition of sera from a number of patients, which I'll be going through the cohort in just a moment. And then from that serum knowledge of that serum composition, matching that up with what we're seeing in terms of our ability to stimulate cells in culture, we did this one. And can we make any deductions about what might be going on with the regulation of signaling in these systems based on those pieces of information? And so just briefly going through our experimental design and how we did this. So we used a model system for this one. We used A549 lung adenocarcinoma cells. We plated them out in near complement cultures, and we ran each experiment in triplicates. We did incubations of 10 minutes total with the patient's sera to allow early signaling events to occur. So that is for the receptor tyrosine kinase itself. And for our patient cohort, we used a total of 134 patients in this one. And really what we wanted to do is cover the spectrum of the disease. That is, looking towards the bottom here, we're covering everything from the high-risk patients. So these are the patients without cancer but are matched in terms of their smoking history. And these are the patients that would have a high likelihood of going on to develop lung cancer. And so they should be relatively matched as far as a control. And then studying the different and having a sampling of each of the different stages. So we're combining stages 2 and 3 because that's where you have the lymph nodes involved. There's little difference other than the extent of progression between those two. But then again, stage 1 is no nodal involvement, and then stage 4, of course, is disseminated. So this really gives us a sampling of the spectrum of the disease so that we can potentially see if there's any differences as the disease is progressing. So after that 10-minute incubation, again, we're running these experiments in triplicate, where each patient sample is sampled 3 times. We'll wash the cells real quickly after that incubation, and we'll create a lysate. And with that lysate, we'll, of course, have protease and phosphatase inhibitors in there to help preserve all the molecules we want to examine. After that, we'll do a BCA assay to get our protein concentrations, and then we're ready to do our assay. And again, here's just to remind you, here's our list of different analytes we're looking at. And we'll use, for this particular one, we used 8.5 micrograms per well, which is about half of what we would use for a single Western blot, just for references. And we're evaluating all these in a single well. And so this is very highly efficient means to do these type studies that this, what happened in a single afternoon in my lab would have taken probably a good month for the same personnel to accomplish doing it via Western blot. So moving on, we had some hits. And the hits that we had definitely are ones that are not unexpected. The hits that we have focus a bit around angiogenesis with Taiwan and EGFR, PDGFR, and then also with energy metabolism with insulin receptor and IGF-1R. And so we're pretty pleased with our initial sets of responses that we've seen in this. And so just to kind of go through a couple examples of these and try to deduce what might be going on. So the simplest case that we saw was with CMET and the HGF receptor. With that, so on the left is the circulating levels, which we actually measured in the patient's sera. And as you see, there's a staircase of these going up, going from the high risk over to the stage 4s, with a couple of notable steps in there where we had significance of, and I'm just labeling that now as a p less than 0.01. And so we've documented that already in a paper that we saw that. And so that was reassuring that we're seeing that again. What was of interest, though, is if you look at the panel on the right, it wasn't until you got to stage 4, the difference between the stage 2, stage 3, so the node positive cases, with the stage 4 cases, that we had any sort of significant changes in the signaling levels. And so again, that's going from the stage 2 and stage 3. Those are the cases where we have positive lymph nodes, and so there's local metastasis, versus stage 4, which is disseminated metastasis. So that's the difference between metastasis in the lymphatic system versus the circulatory system. You can think of it that way. And so that's a very interesting finding. And so what the current study is right now, so again, for these early stage 1s, we're going from high risk to stage 1, and even from stage 1 to stage 2, where we do see these like changes occurring in the HGF levels that are circulating, but there's no changes in the RTK, the ability of those ligands to activate signaling. That must mean that there's something else going on. And so that's the basis of the study, is to see, are there other factors going on? So we're studying that right now by looking to see, are there any decoy receptors potentially available that are circulating that might be sequestering these increased levels to prevent the increase in signaling from occurring. Moving on to another example, so with angiogenesis, there's VEGFR3, which is, again, associated with lymphangiogenesis, so metastatic progression through the lymphatic system. And so on top is VEGF-C, and the bottom is VEGF-D, two of the primary inductive factors for these processes. And not surprisingly, you see with VEGF-C increases as when there's increasing stage. VEGF-D is a little bit more flat as you go across, but certainly when you get to the stage 4 point, it jumps up pretty dramatically. And these are going from the stage 1 up. It's significant for stage for the VEGF-C, versus for VEGF-D, you only have significance at that last step. So very interesting. But what's interesting more so is if you look at the RTK activity, you have an increase going from the stage 1 to stage 2, which would equate pretty well with VEGF-C stimulating more of these cells. When you go from stage 2, 3, so some of the node positive patients to those with disseminated disease, you have a decrease. You have a highly significant decrease. Even though you have a large increase in VEGF-C circulation and a large increase in VEGF-D in circulation. So that must mean there's something else going on. And so that's, again, the crux of what the study is about. And so we're in the process now of looking at more decoy receptors and evaluating by mass spectrometry, are there any changes in the ligand itself? Is there a dipeptidase potentially at play here that might be partially degrading the molecule? And in our final example, I'll talk a little bit about the IGF-1R system. And this is, again, this has to do with the previous example I gave with the fidgetijumab, with the clinical trials not going so well. This is the sort of studies that they probably need to do beforehand, where there's differences in IGF-1 with increasing stage, and even some changes with the binding proteins. But when you look at the RTK activity, there's definitely something else is going on, and something that's not described by even what I'm showing you here, meaning like there's really no changes in RTK activity going on, despite the fact that the IGF-1 levels are changing in the sera, and the binding proteins are changing in the sera. The degrees at which these things are changing should have an inductive effect on IGF-1R signaling, and it does not, from what these studies are showing us. And again, these are done in triplicate, and our box plots are relatively tight here, so I feel pretty confident in this data. The Luminex data, or the data generated by the Luminex platform, for the most part, is very low in CVs, and so it's very reproducible, very highly trustworthy, in my opinion. Anyway, so moving forward, so just to summarize some of the conclusions that I had from today, we use the we're very interested in RTK signaling and how potentially looking at that in the context of patient sera could we better understand some of the processes that are going on with disease progression in lung adenocarcinoma. In the future, we plan to try to redo part of the study using paired patient sera with their with portions of their primary tumors. that might have some interest in terms of, like, are there modifications going on at the receptor level as well? But that's for future studies. We identified in this study a number of different induction of a number of receptor tyrosine kinases with progressing disease. Again, these clustered primarily around the ideas of angiogenesis and energy metabolism. We have a couple examples here where we had an apparent uncoupling of the levels of the circulating levels of these ligands with RTK signaling, indicative of the fact that there's additional regulatory mechanisms going on that have yet to be fully defined, and is something that we're hoping to do in the very near future before we can get this out as a publication. And we saw a similar sort of effect with IGF-1 and the IGF-1R signaling. And again, we believe that this study demonstrates a proof of principle that there's multiple regulatory mechanisms at play, and that, you know, evaluating just circulating levels of a cytokine might be misleading in some cases. And so with that, we're hoping to continue on with this study and try to further elucidate some of these mechanisms. And with that, I'd like to thank everyone for listening to my presentation today, and I'll be happy to take some questions later on in the presentation. I'd like to thank my laboratory staff and some of my collaborators that are listed on the right, and particularly Gabrielle Lobato. She's a graduate student in my lab, and this is a part of her thesis work, and it's very exciting to have her work with us on this study. And with that, I'll turn over the presentation to Dr. Wenrong Li, and she's going to talk about some of her interest in solutions to cancer research. So, Wenrong?
Thank you, Dr. Borgia. That was a great talk on using MILLIPLEX assay to study lung cancer. Thank you. At MILLIPLEX Sigma, and then we have a variety of multiplex panel to explore the hallmarks of cancer. Dr. Borgia has mentioned several of these panels, such as IGF binding protein panel, angiogenesis panel, soluble receptor panel, and the RTK phosphorylation cell signaling panel. In the next 15 minutes, I'm going to tell you a little bit more about the multiplex panel for cancer research highlighted in red. The first panel is the human angiogenesis panel. 1. It can measure the 17 analyte listed here. In this study, human angiogenesis panel 1 was used to quantify serum biomarker expression between normal and cancer sample. In this study, a set of serum samples was used. They were from normal healthy control, colon cancer patient, and also the breast cancer patient. The data on the right demonstrated many of these circulating angiogenesis marker and growth factors are upregulated in the breast cancer or colon cancer serum sample as compared to the sample from the normal healthy control. The same set of serum sample will also evaluate using our human angiogenesis panel 2, which is shown here. Human angiogenesis panel 2 is a 20-plex, detect 20 protein marker, including many soluble growth factor receptor for cancer. And this soluble receptor can potentially serve as the previously mentioned decoy receptors. Shown on the right here of this slide are the 12 circulating protein biomarker from this panel, showing differential expression level between normal and colon or breast cancer serum samples. So up to now, we have talked about immunoassay multiplex panel for human study. For those of you who work on the mouse panel and for work on the mouse model, we also have a variety of mouse multiplex panel for you. These mouse assay kits can help you quantitate multiplex biomarker in different type of mouse sample, such as serum plasma and tumor tissue lysate, as well as the mouse cell culture sample. Next is I will present you an example using mouse MILLIPLEX assay to identify biomarker in mouse tumor for an in vivo model. This example is the mouse Lewis-Long spontaneous metastasis model. High-fat diet was previously shown to enhance metastasis in this animal model. So in this study, two groups of mice were either fed with high-fat diet or low-fat control diet for 7 weeks. These mice were then subcutaneously inoculated with the Lewis lung carcinoma cells. 2 weeks later, the primary tumor from these two groups of mice were resected, and tumor lysae were prepared from the resected primary tumors. We then analyzed the tumor lysae using 4 mouse MILLIPLEX panel as listed at the bottom of this slide: mouse angiogenesis panel, cytokine chemokine panel, mouse metalloproteinase panel, and mouse adipokine panel. As shown in the data panel here, we have identified a putative biomarker with showing differential expression in mouse primary tumor between high-fat diet and the control diet mice. For example, the first one, the MCP1, an inflammatory cytokine, is upregulated in the primary tumor from high-fat diet mice as compared to the control. And the adipokine resistant, the second one, is downregulated in the high-fat diet primary tumor. So we have talked to you about the panel in red, human angiogenesis panel 1, 2, and several mouse panel. Next, I would like to introduce you the emerging panel currently in our pipeline, as shown in here as blue. The first one is human immune checkpoint protein panel, and then the second are 2 BCL2 apoptosis panel. The human immune checkpoint protein panel can measure all of these 15 checkpoint protein in a single well using a small volume of sample. These analyzed in this panel, including the co-stimulating receptors such as CD28, CD27, CD40, and co-inhibitory receptors such as CTLA4, PD1, LAG3, BTLA. Many of these, such as CTLA4, BD1, and PD1 and PDL1, are effective immunotherapy target for improved anti-cancer response. At the bottom, we show you the preliminary data we generate from this panel using the cancer serum sample. We saw higher level of soluble TIM3 protein in breast cancer and colon cancer serum sample versus the normal serum sample. And also a higher level of soluble LAG3 was seen in breast cancer sera as compared to the normal controls. The next panel in the pipeline are the customer-requested BCL2 apoptosis panel. The BCL2 family of protein are important gatekeepers to the apoptotic response. This panel are for the quantification for BCL2 family pro-apoptotic marker and also anti-apoptotic marker, and also the heterodimers. As we can see, the BCL2 family apoptosis panel 1 has the pro-apoptotic protein, BAD, phospho-BAD, BIN, FAX, and 2 heterodimers, BCL-XL and BAD, and also the MCL1 interact with BIN. The heterodimer in this panel 1 can detect the protein-protein interaction of the pro-apoptotic molecule, BAD and BIN, with the anti-apoptotic molecule, BCL-XL and MCL1. The second BCL2 family apoptosis panel is for the quantification of anti-apoptotic protein, BCL2, BCL-XL, MCL1, and the heterodimer of MCL1 and NOSA. The heterodimer, again, is to measure the dynamic balance between MCL1, an anti-apoptotic member, and NOSA, a pro-apoptotic promoted protein. So I just show you a few of the multiplex panel relevant to cancer research. So this slide show you that we currently have over 1,300 different analytes and more than 160 different multiplex panel in 8 different species here. And of course, besides the oncology, our assay application also and go to immunology, inflammation, cell metabolism, cardiovascular diseases, neuroscience, and we have a broad range of cell signaling pathway panels. So in addition to the Luminex platform we talk about today, and our cancer portfolio immunoassay solution also extend beyond to the other platform. So I just want to introduce to you that we have also, besides Luminex, we have the ELISA platform, we have the Gyro Lab workstation, we also have the high sensitivity IRENA system based on the Singulex technology. And we are very excited with the ACMC X probe. That's our upcoming next-gen high-sensitivity assay platform. This single molecule counting assay can help you to detect, for example, the cytokine or growth factor protein marker at the femtogram level. So today we discuss our multiplex portfolio, AR St. Louis site. And we develop and manufacturing a wide variety of multiplex, MILLIPLEX assay. However, if the kits in our portfolio don't fully meet your research need, we also have a custom assay program that can help you create the panel that's right for you. Likewise, our team at Hayward, California, will not only work with you to develop the single molecule counting high sensitivity custom assay, but they will also offer you with ACMC assay services on the IRENA platform. As I mentioned, we are here to design, develop, and manufacture the multiplex assay that meet your research need. So this can be either through our catalog immunoassay panel or the custom assay program. With that, I would like to again thank Dr. Jeff Borgia and his team at Rush University, and thanks to Dr. Lin Yan from USDA for his mouse metastasis model of Louis lung carcinoma. Thanks to our cancer product manager and marketing team at MILLIPLEX Sigma, and my wonderful immunoassay R&D colleagues at St. Louis, Missouri, and also Hayward, California. So please contact us, and we will design and develop the immunoassay solution to get what you want for your research. And lastly, here are our website. You can see the list of all our protein analysis product by visiting our website at www.emdmillipore.com. And for custom assay program, please contact us or send us email directly at customassay@emdmillipore.com. Thank you.
Okay, thank you to you both for that very insightful presentation. I hope our audience found it of great interest and value. As I mentioned at the beginning, if you do have any questions for our speakers, then please continue to submit them using the Q&A tool on the right-hand side of your screen. We have got a number of questions so far, so we'll start addressing them straight away now, live on air. So if I start with you, Jeff, is it possible to evaluate RTK signaling with serum in patient matching specimens?
Absolutely, and actually that's something we're hoping to do in the very near future. We've been experimenting and trying to optimize the system for developing primary cultures for the patients that come through my institution here at Rush University. And by doing that, we can, so I run the biorepository at Rush for the Cancer Center, and so we have excellent access to materials that will permit us to run this experiment as opposed to using a cell line model. We can use the primary tumors. That will allow us to see if there are any changes in receptor that might be also part of the component. Because as I ran it for what I presented today, we're using a single cell line, which might have different regulatory mechanisms in place than individual patients. I don't think we understand well enough yet all the different intricacies of patient-to-patient variation associated with some of these RTK regulatory mechanisms. So yeah, absolutely, that's something we're hoping to do.
Okay, thanks, Jeff. Wenrong, there's one here for you. What if I want to combine biomarkers from different panels?
Oh, okay. Thank you for the question. So at MILLIPLEX and Sigma, we have extensive data on this multiplex analyte compatibility. So if you would like to combine the analyte from different panel, for example, if those analyte are selected from 3 or fewer of our MILLIPLEX panel, our R&D team here, we can work with you, ensure that these analyte are compatible with respect to the, for example, sample dilution, analyte cross-reactivity, or serum metrics if that's required. Then we will either let you know that it is not feasible or it is feasible. If it's feasible, then we will provide you with a special product number, so then you can place the order of that the panel of that will meet your need.
Okay, thank you, Wenrong. We've got a general question. Perhaps both of you could answer. It looks like these kits can be used with cells, tissues, and serum. Can they be used for other sample types?
I'll take that first. Yeah, I mean, if you can put it into a plate, you can measure it for the most part. There's some technical aspects that you would need to cover, and I'd be happy to chat with anyone about that, or I'm sure there's folks at MILLIPLEX that can do this as well. But for the most part, as long as what you're hoping to evaluate is soluble and is something that can be used in any sort of like immunoassay, it can be measured through the Luminex platform and with any of these kits. So yeah, the sky's the limit for the most part.
Okay.
Yes, in terms of sample types, we typically we validate our multiplex kits for serum, plasma, cell tissue, supernatant, and sometimes with lysate. But there are kits that we also specially validate for other type of sample. For example, urine, cell lysate, CSF, and depends on the analyte and the needs. In addition, our kit has been used to numerous publications. So as long as the sample that you can identify, we probably have some reference literature or internal protocol that will be glad to provide that to you and work with you on that.
Thank you very much. Jeff, there's another one for you here. I've heard of some investigators measuring protein biomarkers in urine. Would those have value in studies similar to what you are describing?
Probably for this particular system, not so much. For some of our other studies, they would. But in this case, we're really trying to get at the mechanisms by how RTK signaling might be impacted by different effects, as it exists like in this system. After the process of a protein being excreted involves some proteolysis and some oxidation steps that would have functional consequences for the ligands and the binding proteins too, the so-called decoy receptors. And so we could measure concentrations that way, and certainly there's groups, including our own, which have documented that there's some prognostic utility to those. But as far as a mechanistic study, probably not in this case. So, yeah.
Okay. And Jeff, I've got two more questions. I'll just read them both to you, and then you can answer them one at a time. So the first one is Luminex the best platform to evaluate the effects you documented in your presentation? And the second question is, do you think there would be a difference in evaluating plasma rather than serum in your study?
Okay, so the Luminex part. So yes, I think Luminex is the best, most convenient choice for studying the system as I presented it today. The only other real alternative that I would see that would be cost effective would be mass spectrometry. However, that would require facilities for someone trying to do that beyond what most places have, and but that would be the only other platform that I would think would be capable of it. Luminex, I think, is much more in the hands of the average lab. If anyone's ever performs an ELISA. They, for the most part, can do a Luminex assay with just a couple practice runs. And so I think that that's the best platform for doing this in terms of sensitivity as far as like the different molecules that you want to evaluate. There's a wide range available from Millipore Sigma and some of the other distributors. As far as the second part of that question, which actually, could you repeat the second part of that question? I'm sorry, I lost the track of that.
So it was, do you think there would be a difference in evaluating plasma rather than serum in the study?
Yeah, this is actually similar to the urine question, that in the process of processing the blood, there is some proteolysis involved with the coagulation steps. That could potentially lead to some of the ligands that we're looking at being partially degraded. And certainly that's documented for some ligands, like VEGF-A, for example, can be partially degraded in the plasma preparation, but is not so in serum preparation. Not quite sure, some of the mechanisms involved in, like all the intricacies of the mechanisms, I should say, involved in some of these degradations. But there might be some changes between the two, and really you would have to evaluate to know firsthand whether or not those would be suitable for what you're trying to study. Yeah, so that's the answer to that one.
Okay, thanks, Jeff. One more question has come in here. I saw some of the multiplex data were presented at the past AACR meetings. Do you have more info, such as PDFs of the posters? Perhaps went wrong, or Jeff?
Yeah, because some of the slides I presented today, we presented a slightly earlier version of this at the AACR, this past one, and I can provide those for sure. Probably the best bet is my email information is included in the advertisement for this session. Just send me an email directly. I'd be happy to send you a PDF.
Yeah, and then in terms of the data that I presented today for the multiplex application, two of the emerging area that we are working on, the human immune checkpoint protein panel and also the BCL2 panels, and both were presented with different type of data and also a validation study at this year's AACR. Same as Jeff, will be glad to provide the PDF form of this poster. And if you're interested in that, please just contact us. You can also send us email to or visit our emdmillipore.com /contact, or just call our tech support and or contact us directly. We will provide that to you.
Okay, thank you. We'll move right on to the next question. Do you think decoy receptors are at play in lung cancer only or in other cancers?
I think they're involved in everything, actually, not just cancer. And I think it's, really we haven't had the tools to study this very effectively until recently. And I think now we're at a point with studies like I showed you today, or with other platforms like mass spectrometry, that we can really kind of put side by side what's the concentration of the ligands, what's the concentration of the binding proteins, the so-called decoy receptors, what's the concentration, what's going on in receptor activation, that we can really start seeing like what are the different types of regulation in these systems. And I think it's at play in more than just, certainly more than just lung cancer. In all cancers, I think this is probably at play. But other diseases too, for sure. I'm very sure of it.
Okay, thanks, Jeff. I've got another question here. How do you quantitate RTK activity for the pan-phosphotyrosine markers?
So the readout on this assay is an MFI value. That is, we get a median fluorescent activity, and we do that against the cell lines and all the different conditions that we're testing. But there's also internal controls that are provided by Millipore Sigma for this, and these are several cell lines which they have stimulated, and we use those as references. And we run those multiple times in the assay set in order to make sure that we basically normalize everything to those. So all the values you saw today were from the lung cancer cell lines, but they were normalized to the HeLa cell extracts and the signal that they go through. And so that's the actual figure that's how we kick out, that's how we quantify the RTK activity is relative to a control set.
Okay, thank you, Jeff. I've got one here for you, Wenrong. How many proteins can you measure in a Luminex assay?
So for Luminex assay, we can measure up to 100 analyte in most of our off-the-shelf kits, and then we have larger, for example, like a human cytokine chemokine panel, which is a 41-plex. But we can definitely combine the cytokine, and for example, with angiogenesis or some other selective cancer marker in our circulating cancer biomarker panels, and then we can create a larger panel if that's of interest.
So it has the potential to go up to a 500-plex, if I might just jump in. It has the potential of going up to 500, and we've done 250 in my lab. But you know, as Wenrong was saying, like there's for the commercial kits, there are certain combinations that you have to stick to. Which is fine, because I would encourage people to have very focused questions in doing this. But the platform itself has enormous capabilities.
Yeah, that's correct. Thank you. Yeah. And then for the protein assay, and then since we have to go through the very stringent validation process, and we want to make sure that, you know, there's we maintain the high sensitivity of this assay. And so during the process, so we try to keep that below 100 at this time.
Yeah, that's absolutely critical. I agree.
Mm-hmm. Yeah. Yeah, and I think I also forgot, I just want to add, when I forgot to mention about that the two emergence panels that I mentioned that present at AACR this year, and those are the human immune checkpoint protein panel and the BCL2 apoptosis panel. They are right now, they can serve through our custom assay program. So if any of you are interested in this, and then please also feel free to contact us to our custom program or our technical support program, that we will be glad to help you. Thank you.
Okay, thank you, Wenrong and Jeff. We're going to wrap up shortly, but before we do so, we just have a few closing poll questions for our audience today. So I'll just go ahead and read the first one. Would you recommend this webinar to your colleagues? Please go ahead and select yes or no. Okay, thank you. We'll move on to the second one. What topics would you be interested in hearing about in future webinars? And you can feel free to enter your text in the text box and then click submit. Okay, and then I'll move to the final question for today. Would you like to be contacted by a sales rep? And your options are yes or no. Thank you. Okay, so that's all we have time for today. And as I mentioned before, we'll address all other questions offline. If you do have any questions, therefore, please feel free to send them to us at info@oncology-central.com. That's info@oncology-central.com. I would like to once again thank our wonderful presenters, Dr. Borgia and Dr. Lee, as well as you, our listeners, for your time and questions. If you have a minute, we'd be grateful if the audience members could complete our feedback survey, which you can find in the resource section of the screen. Remember, you'll find a recording of this webinar available to view on demand at Oncology Central, and that's at www.oncology-central.com/webinars, and that will be up very shortly. And that's also where you can find details of our other upcoming webinars from Oncology Central. So thanks again for attending, everybody, and goodbye.